Welcome to the seminar. It is my pleasure to introduce Dr. Wynn Meyer. Dr. Meyer is an assistant professor in the Department of Biological Sciences at Lehigh University. She earned a PhD in population genetics from the University of Chicago. And then she did postdoctoral stays at the University of Pittsburgh and UC Berkeley. Her lab is focused on using patterns of genetic variation to understand how mammals respond to environmental challenges. So with that, I'll leave it to you. Thank you.
Thank you so much, Aldo, and thank you for the opportunity to be here. If you can hear me in the back, could you raise your hand? Okay, thank you so much. So as Aldo mentioned, I come to you here from Lehigh University, which I decided to show on a map in case anyone was not familiar. So here we are in eastern Pennsylvania in the beautiful Lehigh Valley. We are about an hour and a half from New York, maybe 2 hours depending on traffic, and an hour and maybe 15 minutes from Philadelphia. So this does mean that most people in our community are mourning a great loss today. our beloved birds did not deliver last night, but hopefully next year, because Jalen Hurts is just that amazing. And I started at Lehigh in August 2020, which was a very interesting time to start a new job. So I am going to be talking a little bit about some of my work prior to that, which leads into some of the work that I've been doing at Lehigh, which is focused on linking mammalian genomic and dietary evolution. So in the grand scheme of things, I'm very interested in understanding sort of the interplay between genomic evolution and phenotypic evolution. And you can look at this from potentially two different perspectives. So on the one hand, here are our mammalian relatives, and you can see that they occupy a massively diverse array of ecological niches. Mammals have adapted to living in grasslands, in forests, in caves, under the sea. Some of them have even conquered the sky. And I'm really interested in, from sort of a molecular and genetic perspective, how do they do it? What is the genetic diversity that contributes to these massive phenotypic, this beautiful array of phenotypic adaptation? From the other perspective, we have known the human genome sequence for a couple of decades now. We just got a much more complete genome sequence very, very recently. And yet, we still don't know what an awful lot of our genome does, right? We can say, okay, maybe some parts of it encode genes, some parts are potentially regulatory, we don't necessarily know what they are. But from a functional annotation perspective, we're still miles from understanding, if I just pick a random stretch of letters of the genome, what does it do? What consequence does it have for us, for our mammalian relatives, for our fitness, for our health? And what brings these both together for me is convergent evolution. So another way to envision this big picture question is to think about sort of the constraints on how different phenotypes evolve. So imagine you're a mammal in some kind of phenotype space, and maybe you are currently at some kind of less fit phenotype over here, and perhaps it would be advantageous to you to get to this clearly more fit phenotype over here. And I don't know about you, but whenever I drive, I tend to use GPS. However, in the early days of GPS, sometimes the companies that generated these GPS systems, so you can see that there are a few different possible options to do this. Some of the companies that generated GPS directions would give sort of suboptimal results, as shown here. And so what this tells us is that while there may be many possible options, not all of them are accessible to us. We cannot always get there from here. And by studying these things, these patterns of variation in the genome and linking them to phenotypes, we can start to see, are there certain routes that tend to be taken more often, which might tell us something about the level of constraint on the other possible routes. So as I hinted at, The way that I tend to approach this in my work is by studying convergent evolution. And by this, I mean the independent evolution of the same trait in different evolutionary lineages. Here are a few notable examples. So for instance, our human ancestors were not able to digest milk as adults. And in 3 distinct human lineages that domesticated other mammals, they acquired the ability to digest milk using regulatory substitutions at the same gene. Other well-known examples of convergent evolution are, for instance, flight in bird and bat, or the aquatic lifestyle that we see in seals, whales, and manatees. If you think about it, you might be able to guess what their closest extant terrestrial relatives are. Just one second. Okay, so here's the closest living relatives of these guys live at least somewhat on the land. So these are 3 independent transitions to aquatic environment. And cases such as these provide natural replicates for testing how organisms have repeatedly responded to similar environmental challenges. So if I frame the question this way, I can see here the mammalian family tree. This is a subset of the mammalian family tree with a few example convergent traits shown mapped onto it. This convergent evolution provides natural replicates for testing how these species have repeatedly responded to these environmental challenges. What my lab does is it takes, it looks for patterns. So if we repeatedly change the selective pressure on multiple branches of this evolutionary tree, we can see convergent patterns at genes within the networks underlying these adaptive phenotypic changes. And what I mean when I say patterns, it's fairly broad, okay? So some of the patterns that my lab uses are evolutionary rates, which I'll talk about today, gains and losses of function, which I'll also talk about, and several things that I will not talk about, but I hope to study in the future, including the change in size of gene families and signatures of selection within species at the same gene. Okay, so the patterns that I'm talking about, I and others have successfully used these patterns to identify genes and other elements of the genome that have been important for adaptation in 2 different convergent processes, in multiple processes, but a couple that I'll highlight here, specifically adaptation to the marine aquatic environment and adaptation to subterranean environments. So first, I'm going to talk about the marine aquatic environment, which is a project that I was involved with myself when I was a postdoc at University of Pittsburgh, and I have a bit of an update of things that I have worked on since I left. So the specific question that I was asking in this project is a little bit more narrow than what I was talking about before. So it's specifically about functions that have altered fitness consequences in marine aquatic environments. Now, I just talked to you about these 3 distinct evolutionary lineages that have adapted to life in the ocean. We have seals, sea lions, and walruses, or pinnipeds, dolphins and whales, or cetaceans, and manatees and dugongs, which are also called sirenians. Now, they have a number of cool convergent acquisitions of function or changes, like let's say their body types are all kind of large and streamlined, but they also have a number of functions that they appear to have lost in response or along with adaptation to the marine environment. And one notable one, for instance, is olfaction. So if you look at dolphins and whales, you'll see that they've fully lost their olfactory bulb, the part, well, I don't know which part of my brain it is, but it's the part of my brain that allows me to smell, and dolphins and whales don't have it. They've also lost function at most of their olfactory receptor and taste receptor genes. And this is because the ways in which they interact with the chemicals that induce these receptors in a terrestrial environment are totally different in a marine aquatic environment. And they use different kinds of senses when interacting with their surroundings. I can simplify this by saying smell no longer useful underwater. It's a little bit more complicated than that. But while we know these individual examples prior to this project, we had never done a kind of genome-wide scan for this kind of pattern, where each of these lineages has independently lost function at some set of proteins like the olfactory receptors. And there might, for all we knew, there might be other functions that had this shared differential consequence in terrestrial and marine environments. So this is a project that I undertook with assistance of my postdoctoral advisor, Nathan Clark, close collaborator Maria Chikina, grad students Amanda Kowalczyk, Ryan McCarthy, undergrad Clara Crump. And in order to do this, I made use of the fact that we have high quality sequence data for a bunch of different mammalian species, including several marine mammals. And so what I was looking at were these kinds of alignment data, right? So here I have an alignment of genome data. I have annotations from humans as to where the genes are, so I can look specifically at individual coding sequence genes, and I can scan them and score them for substitutions that are likely to cause losses of function in proteins, like premature frame shifts, stop codons, splice site substitution, and mark them with these nice little lightning bolts. Then I can create data for thousands of genes in the genome to say whether they are 1 for functional, 0 for non-functional, X for missing. And I can use a software program to test specifically, if I look at the underlying mammalian phylogeny, whether we see the same rate of evolution from a functional gene to a non-functional gene in marine when the branch transitions to an aquatic environment as when the branch is not transitioning to an aquatic environment. So this is just a standard likelihood ratio test implemented in the program Bayes traits. And when I run it on some over 10,000 genes genome-wide, what I see is some of the usual suspects, okay? I expected to see olfactory receptors, I expected to see taste receptors, because we already know those have convergently lost function in marine aquatic lineages. What I 100% did not expect to see is this top gene called peroxidase 1, which I was very curious about. So in your body and in mine, you would find PON1 in your bloodstream associated with HDL and LDL molecules. And specifically what it does is reverses oxidative damage to lipids on these HDL and LDL molecules. Now, via a similar chemical process, PON1 also can metabolize toxic oxon byproducts of a specific class of pesticides called organophosphates. And this is important because if you or I were working in an agricultural setting for long periods of time and were exposed to these toxic oxon byproducts, we could experience organophosphate poisoning, which has a range of symptoms running from shortness of breath to coma and even death. Now, we thought that this particular phenotype might have negative consequences for some marine aquatic species that might encounter these organophosphate pesticides in their environments. And so we undertook a further confirmation of our findings by looking for the action of the PON1 enzyme against its organophosphate substrate. And we did this in collaboration with a number of wildlife biologists who provided us with samples, as well as biomedical chemists who could run the assays to test for the metabolism of these organophosphate substrates. And what we see when we look at this is that, first of all, this is the level of activity against a specific organophosphate substrate. Here's the level of activity, the standard level of activity for a knockout mouse. You can see this is what happens when you don't have the enzyme functional. And there's a range of activity for terrestrial mammals, which are labeled in these colors here. But what you can see is that for the blue aquatic species, most of them have either near zero or zero activity against these organophosphate substrates. So my collaborator, Stacy Woods, made a sort of envisioning of how this might affect species like the manatees, whose environment shown in this bright orange color abut agricultural land that specifically uses organophosphates in this kind of green color overlapping their waterways and parks. So this is a bit concerning. But from an evolutionary standpoint, we were also interested in when and how often this enzyme lost function. So with some of the collaborators in this project, we looked at the rate of evolution of coding sequence in this gene on different branches of the tree and used some math to calculate when the non-synonymous to synonymous substitution rate went from a background constrained rate to a rate closer to 1, which is what happens when you're a pseudogene and it is no longer detrimental for you to accumulate non-synonymous changes. So we use that method to estimate that PON1 loss function very, very long ago in the ancestors of manatees and dugongs and in the ancestors of cetaceans, but much, much more recently in the ancestors of seals and sea lions or pinnipeds. And in fact, more recently, through further study, we have found that there are in fact multiple much more recent losses in these semi-aquatic lineages if we add more data for additional pinniped species. Okay, so with collaborators, I'm continuing to investigate the process by which PON1 loses function in semi-aquatic mammals, in part to predict risks that these extant species might have in the presence of organophosphate pesticides, and in part to try and understand how and why PON1 may have lost function in the past. Now, this is an ongoing project co-led by myself and Ali Graham, postdoc at the University of Utah. When I was at Pitt, Jerica Jameson, who was then an undergrad, was leading the charge on this with help from Lexi Michaels and Charlotte Patrick. So based on some additional Sanger sequencing, some additional enzymatic assays that I'm not going to show you all the data from today, we jointly inferred that PON1 has lost function at least 3 independent times in the ancestors of extant carnivorans. So that's here we have some seals and sea lions up here, and here we have some sea otters down here. This was an enormous amount of work, especially by Ali, in collecting additional samples from multiple semi-aquatic species. And one thing that I'm particularly interested in is understanding sort of how PON1 has lost its function. So here's one particularly interesting example where we see that the functional loss appears to have occurred on the ancestral branch of these extant species. However, if we look at the specific changes that let us know that PON1 is not functional, there aren't any that are shared. So this species has a nonsense and splice site substitution. This species has a different nonsense substitution. And this one does not appear to have any protein coding sequence changes that would predict a complete loss of function. However, when we do those enzymatic assays for many, many samples within the species, we never see any plasma activity. So what this hints at is that there may be some kind of regulatory substitution underlying this shared loss in these species. And students and I are doing some additional computational work to try and uncover, to see if we can identify what kinds of changes might be driving this. Another question that we were very interested in answering is why PON1 function might have been lost in aquatic and semi-aquatic mammals. And in our original study, we formulated 2 different hypotheses about this. So one, and they have to do with PON1's known function, right? So we know that it's a pleiotropic enzyme, right? So it works with oxidized lipids in the bloodstream, and it has these impacts on our ability to detoxify organophosphate substrate. We know that organophosphates were not involved in the initial loss of PON1 because they didn't exist 50 million years ago when PON1 was lost in the ancestors of whales. Right? So we have 2 different hypotheses. The first is perhaps this loss has something to do with a shift in lipid metabolism. And so what is being shown here is the balance of omega-3 and omega-6 fatty acids in the diets of different semi-aquatic species, from marine to terrestrial. And so since these two different fatty acids have different propensities for being oxidized, we thought that perhaps this could explain PON1's loss. And we wanted to look at information about which species have lost PON1 and what the contents of their diet were to see if we could do an association with PON1 loss and diet. Here's where diet comes into the marine story. Okay. Another hypothesis that we have is that the loss might have something to do with diving. So species that spend much of their lives underwater have something called a dive response. And one thing that you notice if you look at the sequences of their antioxidant proteins is that there have been massive changes to both the sequences and the levels of antioxidants in their bloodstream. So since PON1 is involved in this antioxidant process, we think that perhaps it either became obsolete or was in fact detrimental to habit because of this antioxidant rewiring involved in the dive, the evolution of the dive response. So to test this, Jill used a phylogenetic logistic regression to compare the functionality or non-functionality of PON1 with two different measures representing these two distinct hypotheses. So here is an estimate of the omega-6, omega-3 fatty acid ratio in the diets. And these are diets that are slightly more similar to terrestrial mammals. And these are diets that are slightly more similar to aquatic mammals. These are estimated maximum dive times from observational studies. And again, here are shorter dive times and here are longer dive. So what you can see is that in the diet, the direction of the association is sort of in the opposite of what we would expect, where PON1 tends to be more functional when diets become more similar to marine aquatic. And in the dive time data, it is in the direction that we would expect. Okay, so more terrestrial, shorter dive times predict more PON1 functionality, longer dive times predict less PON1 functionality. What kind of arthropod have we got in here? Ooh. Oh my God. Someone's study subject? I don't know. Don't read it. Just okay. So we have a fairly small sample size. We have some phylogenetic correlation in here. So it's kind of challenging to do this test with much power. However, it appears to be the case that our hypothesis related to diving seems a little bit. So far, what we've identified is that PON1 is the most striking signature of convergent functional loss in the entire set of coding sequences for which we have alignments in mammals. We know that this functional loss occurred shortly after the aquatic transition in multiple polyaquatic and semi-aquatic lineages. And we know that it occurred at least 6 times in different semi-aquatic species of mammals. I talked about the cetaceans, sirenians, and pinnipeds. We also see evidence for functional loss in beavers. And we think that it's possible that this functional loss might be due to some kind of something involved in the dive response. And it's likely not related to diet, but we don't know exactly, we still can't answer exactly why. And based on, sorry, this watch is very insistent. Based on the evidence that we have continued to collect, we also see in some of these lineages that have not lost their protein function, they do appear to have reduced expression if we look at publicly available RNA sequencing data for these species. So what this suggests to us is that perhaps there was some kind of the process by which PON1 function appears to be lost is that initially there's some kind of substitution that influences the regulation of the PON1 gene, which leads to reduced expression, reduced protein levels, and that allows changes to accumulate in the PON1 protein, which shows evidence at the level of sort of non-synonymous to synonymous changes, further reducing the protein level. And then eventually you have substitutions that create a non-functional protein. And these are sort of the spots at which we think these different organisms are in this process, with the pinnipeds sort of representing a range throughout this entire process and enabling us to further investigate and see what kinds of regulatory changes might be enabling this stepwise process towards loss of function. So, as I mentioned, this project began when I was at the University of Pittsburgh with Nathan Clark and Ray Chiquina, who are really delightful collaborators. Nathan is now at the University of Utah, where his postdoc, Ali, has been leading the charge on this. But I just wanted to acknowledge all of the people who have been involved in this process. I was telling someone earlier today that one of the things that I really love about science is the opportunity to collaborate with people from such a wide variety of different fields whose different expertise contribute to make such a fuller story of something that otherwise I would just have a very, very narrow window into. So I am exceptionally grateful to all the people involved in this project. So building on this great success story, where we identified something that we had never known before about marine aquatic adaptation in mammals, I undertook my own independent research program with one of my goals being to identify the molecular basis of adaptation to dietary specialization in mammals. So here I have a cartoon of the mammalian family tree where the colors and icons represent sort of a simplification of each of these species diets, with green being herbivore, blue omnivore, and red carnivore, or orange insectivore. What you can clearly see from this little cartoon is that several of these phenotypes are convergent, right? My favorite kind of phenotype. And this repeated adaptation enables me to use these patterns that I talked about before to identify genes that are important for the same diet in different evolutionary lineages. Okay. I'm going to show a little bit of a cartoon of the molecular evolution patterns that I'm going to be looking for in this section of the talk. So apologies if this is a review for many of you, but the way that I tend to represent this is by way of trees, right? Where branch length in these trees represent accumulation of either nucleotide or amino acid substitution. Now, each gene has its own tree, but let's say this is what we would call a typical gene. Most genes in the genome are under sort of some level of purifying selection. So the branch lengths represent the neutral-ish substitutions that are able to be accumulated over time. I'm going to zoom in on one branch, and I'm going to ask what might happen to this tree if the degree of selective pressure were to change. So here we have selective pressure experienced by a typical gene. If we increase the level of purifying selection such that substitutions on this lineage are no longer quite as permissible, then what would happen to this branch? Okay, so for a branch, thank you. So when we increase purifying selection specific to the cow lineage, right? Like let's say this gene is particularly constrained within the cow, then we see that the cow branch specifically is shorter. In contrast, if we have a relaxation of purifying selection, something like the olfactory receptors in cetaceans, for instance, then instead we would see a lengthening specifically of this branch. Now we might see similar patterns under some models of positive selection as well, this gets a little bit complicated, so I might come back to it. But the power of convergent evolution is that it allows us to look for these patterns on not just one, but many branches at once. And so this led to the development of this software tool called RAR Converge, which I am happy to promote, having written a lot of documentation for it. And it uses these specific patterns to identify genes that have experienced different selective pressures in different environments. So let's say we have selected genes or relapse selection genes that have longer branches, accelerated evolution in these green foreground species. And now let's imagine that these foreground species represent mammals that have plant-based diets. If we compare the length of these branches to those of that expected gene, right? So we can average these branch lengths over all genes in the genome, we get an average or expected gene length. And then we can compare the length of this branch for each of these foreground lineages to what you would expect based on the genome-wide rates of evolution, right? So here we have this specific foreground branch, and it's rather far from the expected x equals y line. So we can use the corrected residuals from this to provide a quantitative estimate of the relative rate of evolution on these branches relative to the expectation under standard purifying selection. So now that we have this standardized relative rate, we can compare it between our green foreground branches and our black background branches, and then we just have a test for, does evolution appear to be accelerated specifically on the lineages that have experienced this environmental shift. And so my lab has used this to undertake an investigation of convergent dietary adaptation using newly available genome data for a large set of mammals. Now, first, we wanted to define the phenotype in a very consistent way. And so my grad student, Katie Foley, and several undergrads, including Kumi Ojo, pictured here, performed an extensive literature search to identify mammals with dietary extremes. So we're looking for sort of pure carnivores versus pure herbivores, where the species have more than 50% of their diet made up of vertebrates, or more than 50% of their diets made up of vegetation. And we combine this phenotype data with molecular data from the Zoonomia Consortium, which is a massive, again, somewhat interdisciplinary group working to combine genomes from many species of mammals into usable data sets for comparative genomics analysis. So the alignments were made with this program called TOGA, which Michael Fuller's group developed. I filtered these alignments to retain high quality data. And we focused on a subset of mammals for which there was actually variation in these extreme diets. Because if you look at some sets of the tree, there's not really phenotypic variation, and that does not provide statistical power to associate genotype with phenotype. I estimated these gene trees, fixing the topology or the shape of the phylogeny based on the species tree. And then this resulted in about 15,000 genes that we also filtered to include only those with a large enough number of species in the alignment that for which we had power to detect these associations. As an additional step, we tried to correct for the fact that these phenotypes show up in the tree in this kind of clustered manner. So we have a whole bunch of branches of related species that are carnivores, and a whole bunch of branches of related species that are herbivores. And sometimes evolutionary rates tend to be correlated within certain subsections of the tree for reasons that have nothing to do with the phenotype. So to correct for this, the developers of our RAR Converge have developed a really cool tool called permulations, which is a combination of simulation and permutation. And what we do is that we use phylogenetic simulations to generate matched trees that kind of have similar numbers of foreground branches, but they're not the same branches as what we see in our real trees. And this is a way of correcting for phylogenetic non-independence in the data. So you can see a couple of examples of these matched null trees that we generated through this process. So one challenge that I've been facing in analyzing these results is that once I do these kinds of corrections, I seem to not have as strong of an enrichment of true signal in the results. So maybe it's a little bit challenging to see this, but if you look at, for instance, the purple corrected permutation-based p-values, without the correction, it looks very striking. It's like, okay, we have lots of genes that are potentially associated with diet, but once we correct using permutations, the signal seems to go away. So one of the things that I think might be happening is that because we have quite a lot of branches that are that have the same phenotype, it makes it really challenging to create these null sets that are perfectly matched. And so one of the things that I'm hoping to do in the future is to prune the tree a little bit and see how robust the pruning is to different subsets of the tree. Nevertheless, there are still a few genes that reach genome-wide significance with this correction factor, and one of them is this one called CISD1. One thing that I found is that the genes that survive this correction process tend to have long branches in the transitions. So here's like an ancestral branch leading to a whole bunch of herbivore branches. Here's another long ancestral branch. And I think those kinds of things are more robust to the permutation process. But what you can see here is sort of the distribution of relative rates for pure carnivore versus pure herbivore branches. This, I should mention, is work by former grad student Katie Foley, current grad student Michael Tene. And one thing that I'm very excited about this particular gene is that it interacts with iron, which is an element that is in different proportions based on species diet. Another thing that we can do beyond looking at individual genes in these outcomes is to look for gene set enrichment. And one thing that I'm particularly interested in doing is looking for enrichment by the tissue in which the genes are expressed. So in order to do this, I used a publicly available expression data set from humans called GTEx. This is available for nearly 1,000 humans. They have RNA sequencing data for 52 different tissues. And then I assigned genes to tissues not based on tissue specificity, but just based on which tissues they were most highly expressed in. So I have transcripts per million for all of these 52 tissues, and I find sort of the place at which there seems to be a gap between high expression and low expression tissues. And then I just assign the genes to all of the high expression tissues. So when after I do this, then I'm going to show you some results, and the sign of those results is based on the direction of the association. So genes expressed in that tissue that evolve faster in herbivores have a negative sign. And the test statistic is for a difference in the distribution of the gene-specific association for the tissue-expressed genes versus all of the genes. So here are some of my results for the tissue expression pattern. If your favorite tissue is on here, please let me know if you want to have a look at which genes evolve differently based on diet. And I'm happy to talk about potential follow-on experiments or even just potential further bioinformatic tests to see if there are specific kinds of patterns driving these signals. One thing that stood out to me is the involvement of genes that are expressed in the brain and in the thyroid. which seem to be evolving much more rapidly in herbivores than in carnivores. There are a few other expression patterns. I suspect the testis pattern is noise for some reasons that I'm happy to talk about. But there are several other tissues that I think either might be involved in adaptation to diet or might correlate with phenotypes that are related to diet, like various life history traits. So this is a work in progress. I just want to walk through some of the next steps that I have planned, and I am also really open to ideas and suggestions. So from an analytical perspective, what I'd really like to do is explore a number of other dietary phenotypes, some of which potentially don't have this problem of being highly phylogenetically correlated in their foreground branches. I'm also really interested in defining diet in a quantitative way so that I'm not just looking at carnivores versus herbivores, but probably also looking at amount of sugar in an animal's diet, amount of lipids, degree to which the animal grazes versus browses, and various other things like that. Open to suggestions on this front as well. I am looking at various pipelines to identify the number of copies of gene families in different organisms' genomes. And I am interested in correlating that, the gene family evolution with dietary evolution as well, and to see which genes seem to contribute to dietary specialization at the single gene level versus the gene family level. I'm also interested in using the large amount of genomic data that we have for extant humans to try and identify signatures of selection in human populations that are relevant to dietary phenotypes, and to compare which types of genes and regulatory changes we see at this very short evolutionary timescale versus the kinds of things that we see if we look at a much deeper evolutionary timescale. And then the thing that I had really hoped to have an answer for you today was looking at differential expression. So looking at protein coding evolution versus patterns of gene expression evolution in relevant tissues to dietary adaptation, and seeing which genes tend to evolve via protein coding substitution versus patterns of expression change. And I have some publicly available sequence data for this. I would be really excited to generate more data for more species, for more tissues. Any of you know of ongoing efforts in that vein, I'd be really happy to work with them. Okay, on the experimental side, this top gene, and along with a few other genes, we have known structures for them. And so one thing I'm really interested in doing is seeing which parts of the protein seem to be contributing to this signal of differential evolutionary rates between, say, carnivores and herbivores. And I have a biochemist friend who is excited to make different versions of these and to look at what kinds of consequences that might have for the protein structure. I also have a yeast collaborator, and one of the sets of genes that came up in my analysis, yes, yeasts are awesome. One of the sets of genes that came up in my analysis was branched chain amino acid gene. And so what I'm interested in testing is whether different orthologs from different species might have different consequences for metabolism of, for instance, branched chain amino acids if we test them in a yeast model. So like I said, this is a work in progress. My work on connecting this coding sequence change to potential change in protein structure and hence protein function is a collaboration with the Fisher Lab in the chemistry department involving a couple of undergrads, David and Matt. I have a grad student, Josh, and an undergrad, Anya, working on identifying different sources of quantitative dietary phenotypes, which I hope to summarize in a way that is less confusing to explain than this. But this is kind of your standard PCA for different measures of measurable phenotypes that might be related to diet, like microbiome diversity, trophic level, and presence or absence of different organisms in cells. And then on a completely different note, I am working with a grad student, Rachel, undergrad Leah, and members of the community to identify methods for exploring and understanding the genome of our local invasive pest, beautiful local invasive pest, the spotted lantern fly, to get people excited about the prospects for using genomics as a tool to understand and improve our environments. So before I wrap up, I just want to take a quick detour to talk about an exciting experience that I had outside the academy. In the 2019 to 2020 school year, I was a AAAS Science and Technology Policy Fellow in the executive branch, and I worked in a teeny office that no one has ever heard of at the NIH. This is my cohort back in fall 2019. How little we knew standing in front of the Capitol. But I just wanted to give a few unsolicited thoughts about my time working in the federal government. None of these are approved by my institution or my office. So first off, one thing that I noticed is that we often talk about the government does this or the government does that, or we hate that the government is blah, blah, blah. We know this is a simplification, but spending a year in DC made that ever the more apparent. There are a ton of different agencies within the government, some of which I had never heard of. And one thing I appreciated from working in a bipartisan office is that, especially within some parts of the executive branch, most people there are public servants who work there for their entire lives under administrations of many different political flavors. And it's much more about the mission than about the politics, which was really reassuring to me. Additionally, when I interviewed at a wide variety of different agencies. In addition to the NIH, the NSF, the USDA, the Department of Defense, Department of Energy. I had lots of friends who worked in the State Department, and in all of these different subsets of government, the agencies really valued and respected the scientific expertise of my friends in the fellowship and the full-time scientists working in these agencies. So I think a lot of times we don't necessarily see that. And again, this was reassuring to me. Another thing that I didn't really expect, but grew to appreciate, is how challenging it is to get feedback as a government agency from the public. They really want to, but it's hard. So there are a couple of ways for you, for me, for anyone, to provide feedback. Different agencies ask in very obscure ways, but you can always find things that you can publicly comment on, and they are by law required to read the public comments. So one sort of clearinghouse is regulations.gov. NIH has one specifically. It's the same search guide you use to search for funding opportunities. They also have these things called requests for information, or RFIs. And there's one open right now through the middle of March that I think people might actually have thoughts on, which is on the review criteria for NIH grants. They're simplifying the review criteria, sort of in an attempt to reduce the emphasis on prestige of institution. Sorry if I got too political, y'all. So yeah, I think I'm also happy to talk about anything else, any other questions you have about my year working in the Trump administration. But yeah, I had a grand time, and I met some amazing people and would recommend it to anyone. So with that, I just want to wrap by acknowledging all the folks who are continuing to contribute to the success of my lab, especially my very first graduate student, Katie Foley, who is about to be a successful attorney. She's going to graduate from the University of Iowa College of Law pretty soon, which is fantastic. my new grad student, Michael Tenney, who is driving this convergent evolution in mammal diets forward, and a whole crop of amazing undergraduates. Ongoing collaborations in this area, particularly with Irene Kaplow at Carnegie Mellon and Emily Pinkett at University of Memphis. New funding from NSF, which is allowing me to continue this work. And I really want to thank Rebecca and Aldo for making it possible for me to be here today, and you for your patience. I also just want to thank everybody who's made our lives livable over the past few years. The past few years have been a lot, right? So especially for sort of frontline workers, for all the researchers who made some of our healthcare possible, and for anyone in the community who's been involved in parenting, teaching, childcare, any of those things, you are all freaking superheroes. And just the people who've been supporting each other during these challenging times. Thank you so much.
There's time for a couple quick questions. Thanks for your talk. For the first part of the results, I wonder if you could talk a little bit about what regarding the parent positive branch length or positive evolution in the primate lineage. It's
Yeah.
a little out of the scope.
Yeah, let me see if I can. So I might fail to answer your question, but answer another related one. You see, wait, no. Where did my Zoom go? Okay. Can Zoom people see this or only the? Okay.
Yeah, so it's about those orange branches, basically. I think those are given, and I know it's kind of not.
Yeah, I think so. I think part of this has to do with the way that the tree is pruned and the fact that the branches are short. I haven't investigated that quite as much, but I can. One thing I have investigated is that there are some really interesting patterns going on in the bats. And we think that might also have something to do with their differentially hypoxic environment. But the kinds of changes that we see in bats are different from the kinds of changes that we see in the marine mammals. There's probably something really interesting going on there, and we're not quite sure what it is yet. But thanks for asking. Yeah.
So if I understood correctly, like the comparisons that led you to the CISD1 gene, you chose an optimal subset of mammals that would give you the diversity at trophic level you needed to find that. Once you find genes using that strategy, do the patterns hold if you look more broadly then?
choose a different subset?
Or just, yeah, if you look beyond this
okay.
species, then are there still interesting, is this gene still doing interesting things in other species outside of this tree?
Okay, so if I understand your question correctly, you're saying, does it matter that I chose this specific subset of mammals? If I chose a broader set of mammalian species, what might this gene be doing? What might the evolutionary patterns look like at a wider, with a wider
I guess
scope?
not so much of a technical question. It's like, do the patterns you find in your subset hold beyond this subset of species?
I think that's a great question to explore, and I think one particular challenge for this phenotype is, like I said, it's not necessarily variable outside this clade. So then the question is, if this is relaxation of selective pressure, particularly when species have newly evolved herbivorous diets, do I see it in species that have been, for instance, herbivores for longer in some of the other clades? Or what kinds of patterns of evolution do I see if I look at this in parts of the tree that are not phenotypically variable? I'm not sure what to predict, and I haven't done that, but I think it's a reasonable thing to do. I don't know if that's actually what you were getting at. Can I answer a quick question in the chat? And then I'm sorry I ran long. Felt like I was stalling. Jeff asked, maybe this falls under your patterns of selection within humans category. He said he's wondering about the gene phenotypic connections that I identify from interspecific evolution. Can this gene phenotype information be used to usefully predict phenotypic consequences of human SNPs? Yes, in some cases, yes. And so it's Nathan's work, but the things that he has been working on in subterranean mammals have identified regulatory regions that appear to become sort of jumbled in independent mole lineages that live underground. And he's been working with some people with human eye disease data to see whether he can draw connections between sort of human patterns of variation and predictive power for sort of functional consequences for changes in those regions in humans. And so far, that's looking very promising. I can't say for some of the other phenotypes, there's not as clear of a parallel with the marine species, but I'm very interested to see whether we can find anything predictive with the dietary types. Yeah.
Thank you.
So we have a finite set of genes.
Yeah.
And I'm wondering if you can identify genes or find some genes that where the organisms can't afford to jettison the coding region, but they can shut it down in certain tissues. If that's going to come out of your gene expression tissue analysis.
Yeah, so I think the question is, are there some situations in which the level of evolutionary constraint on the protein coding gene is such that we can't really see these convergent patterns if we just look at protein coding genes, but we might see something interesting if we focus our attention on tissue-specific regulatory elements, or potentially just expression. I am sure this will be the case, and I am hoping to work on that. I'm not there yet. I have some collaborators that are working with me specifically on tissue expression data for liver, which is a tissue that's available for a whole bunch of different species. And we're interested in sort of, we have a few potential tools that we want to use to correct for phylogeny when looking for differential expression in liver. And we're interested in kind of looking at the overlap between genes that evolve rapidly at the coding sequence level versus genes that evolve different gene expression patterns. And we have a couple of different tools that we're also hoping to use to look for regulatory elements whose sequence patterns might be indicative of lineage-specific changes that might be driving these different expression patterns. It's a lot harder than coding sequence analyses because there are so many more of them. They're not so easy to annotate and align. There are all kinds of, they're short, right? So there's not as much information about the rates of evolution. But we have a few tools and we're working on that, and I think that's an awesome and exciting question.
So I had a question. So in this example, the change in diet is really drastic, right? It's carnivore versus herbivore. Do you know if some of these patterns will hold when you look more intermediate diet effects, say omnivore to either a carnivore or herbivore? You would still expect those long branches in that shift, or if that effect is going to be smaller.
So the question is, will this gene show up, or would other patterns hold if I look at slightly more subtle phenotype changes that are not just pure carnivore versus pure herbivore? I actually think that once I start looking at quantitative traits, my power will increase because I can include more species. And I think it's likely that there are some changes that are going to be persistent across these more subtle shifts. So I'm really excited to dig deeper, to get the phenotypes more clearly defined and to be able to include more of these subtler transitions. I don't know if that answered your question.
Yeah, I mean, so we don't know yet. Then what?
No, I want to do it. Let's do it. Yeah.
You might have mentioned this, but so is the idea behind like all the independent PON1 non-functional genes in seals, is that because of like you have a better ability to detect it because it's more recent? Like Or like, do you think that like that process is happening, like that you have independent changes and like cetaceans or something is just about like what you've sampled? Or is like there one independent loss of function on one that all cetaceans have, or that the
Okay, so this is an interesting question. I like this. The question is, do you think that you see all these independent losses of function in your seals because you're catching it more recently, and you would have seen a whole bunch of independent losses of function in the cetaceans, but it's sort of wiped out by the fact that it's far in the past? Or is there something else going on specifically in the seals?
or like about your sequence of the cetaceans or
Yeah. So what I didn't show you is that all of the cetaceans have multiple shared loss of function sites. So if you look at like all extant cetaceans, they have similar sort of patterns of non-synonymous or nonsense substitutions, right? And same for the manatees and dugongs. They have at least one shared substitution. And we don't see that. We do see that here, we don't see that here. So I don't know whether that's like they all had multiple substitutions, but they also had some shared ones, and it happened so long ago that we can't tell. I do think that because this is very recent, it allows us sort of more fine tuning for understanding